Hi, student. Good morning. Today we are going to discuss topic number 5 of IGCSE biology, and the title is enzyme. Okay, so firstly, the question arises in our mind that what the enzyme is. So that enzyme, are the biological catalyst, okay, which act as a speeding up agent, okay, and these enzyme, uh, speed up the reaction without being using it. Okay, so see here, enzyme are protein that act as a biological catalyst. A catalyst is a substance which is speed up a rate of reaction in chemical reactions without being used up. The enzyme is not changed by the reaction and can be used repeatedly. Enzymes are crucial to living organisms as they speed up metabolic reactions which would take too long to occur without them. So here, you should know the difference between the enzyme and catalyst. Catalyst is any agent that you are using in the chemical laboratory, okay, that speed up the chemical reaction, okay, and it is doesn't use in it. But catalysts, catalysts are also working in human body, which is speeding up the chemical reaction. But these type of catalysts we named as enzyme. So enzyme, are the biological catalyst, okay, that speed up the chemical reaction. Because in our body, so many metabolic reactions are taking place. Okay, for example, respiration, okay, and like electrical impulses, transfer from one place to another place. Okay, so many compounds are being made in the body. So many compounds are being broken in the body, photosynthesis, in plant. Okay, so all these chemical reaction, requires enzyme that speed up the chemical reaction. Okay, now see enzyme action, how the enzyme works. If you are going to study the structure of the enzyme, you'll see that enzyme, has a specific site that is called as the active site, where, like, the specific substrate is going to be attached, okay, and then it's going to make a enzyme substrate complex. After that, it's going to be broken down. Okay, so in each enzyme is complementary to only one type of substrate. Why? Because the shape of the active site, is a specific. Okay, so this shape, is this substrate shape, is going to be fixed into the active site. Then it is to be broken down. Okay, then enzyme can work on it. This is also called as the specificity of enzyme. Why? Because a specific enzyme, is there for a specific substrate due to the specific shape of the active site. Okay, as you can see in the diagram, see, this is enzyme 1. Okay, enzyme 1 is complementary in shape to the substrate, will catalyze the reaction. Okay, but see, if this substrate, that is, you can see here the yellow color, okay, is if it's going to be fixed in the enzyme 2, it will not fix. Why? Because the shape of the active site, okay, is different. Okay, so this substrate is to specific to enzyme 1. So this substrate is only fixed for the enzyme 1. Okay, enzyme 2, has a different shape. Okay, so it will not, the substrate will not fix in it. Now you can see here that, the substrate is there, okay, and the active site is there. Okay, you can see here number 1, okay, the enzyme substrate complex. After that, this substrate, is going to be fixed here, so it's going to make enzyme substrate complex. After that, you can see that the enzyme breaking down the substrate. You can see the substrate is broken down. Okay, substrate is broken down, and you can see the products here. Okay, after that, this enzyme is again ready to do an other catalytic reactions. Okay, now see enzyme specificity. As I've told you that each enzyme, has a specified shape of the active site. So see, enzyme are specific to one or particular substrate as the active site on the enzyme where the substrate attaches. Is a complementary shape to the substrate. This is because the enzyme is a protein and has a specific 3D shape. its shape is 3D. This is known as lock and key model, lock and key hypothesis or model. So, this enzyme, work as a lock, and the substrate work as a key. As you know, you cannot open the lock by any of the key. In the same way, any substrate cannot be broken down by a specific enzyme. So each enzyme, has a specific substrate. Okay, when the substrate is going to be fixed in the enzyme in such a way that as like the key is fixed into the lock, okay, then the door can be opened. In the same way, the substrate can be broken down if it's going to be fixed into the active site of enzyme. When the substrate move into the enzyme's active site, they become known as the enzyme substrate complex. After the reaction has occurred, the products leave the enzyme active site as they no longer fit it, and it is free to take up another substrate. Okay, now which factors are responsible to increase or decrease the activity enzyme? So here in IGCSE, basically there are two factors, pH and temperature. If you are going to study like the other like courses, okay, especially if you are going to study like the A level, so there are also two more like factors are there, like the concentration of the enzyme and the concentration of the substrate. Okay, but here we have to just discuss only like these two factors, pH and temperature. Now the question arises, what the pH is. PH is the concentration of hydrogen ion. Okay, and this based on the pH we just say that it is a neutral solution, it's acidic, it's a basic. If the pH is 7, so it is neutral. If more than 7, it is alkaline, and less than 7, it is acidic. So enzyme have an optimum pH that they work best at. I mean, each enzyme has a specific pH where it work best. For example, if you are studying the pH of the stomach, the stomach pH is about 2.2. Okay, at this pH the protease work best. Okay, so protease act on protein and it converts this protein into polypeptides or amino acids. Okay, but this pepsin or protease acts at pH 2.2, that is acidic pH. As the pH move away from this, the rate of reaction decrease. The change in the pH causes the shape of the active site to change. This means that the substrates cannot fit in the active site. Hence, no enzyme substrate complex can form. This means that the enzyme has become denatured. Yes. If like, the enzyme is not going to take the place to substrates to be attached, okay, so this reaction will not proceed and enzyme substrate complex will not form, which means that the enzyme has become denatured. Now, another, factor, is temperature. Okay, so temperature, as the temperature increases up to the enzyme optimum, the rate of reaction increases. Okay, this is because the molecules have more kinetic energy, thus move faster. Consequently, more successful collisions occurs and more enzyme substrate complex can form at a very high temperature above the optimum. The enzyme become denatured. And the active site change shape. This decreases the rate of reaction as the enzyme substrate complexes cannot form. So you know that if like the temperature is given more than the optimum point, okay, so the active site of the enzyme is D-shaped, okay, or its shape is changed, due to which the enzyme is going to be denatured or damaged, okay, and the reaction will not proceed. For example, like the optimum temperature for working with human enzyme is 37 centigrade. If the temperature increases like 38 or 39, okay, so the enzyme are slow down means their activity of enzyme slow down means all the reaction are going to be slowed down. Okay, and if the temperature reach like or like reach to the 40 or more than that, okay, so the enzyme are going to be denatured or damaged. Okay, and once the enzyme denatured or damaged, it cannot reback again to the active activity mode. Okay, you can see here This is, this is like, amino acid chains making up the enzyme. This is, you see, the enzyme is there, and this is the active, this is the substrate, and this is the active site. Forces between the amino acids in the chain hold the enzymes together and create its unique shape. Okay, because what are enzyme? As we already discussed that all enzymes, are protein in nature, and protein means different amino acids are there. So different amino acids make a specific shape for the active site. So any substrate that's going to have the resemblance with the shape of the active site is going to be fixed. Okay. So when it's going to be fixed, then it's going to be broken down. Okay. So increase in the temperature. For example, like, the temperature that is required for this reaction. Okay. It just crossed the optimum limit. So what happens? See, the shape of the active site is changed. Once the shape of the active site is changed, then this substrate cannot attach with it. So the reaction will not proceed. Okay. And we say that the enzyme is denatured or damaged. Here you can see another diagram. See, the substrate is there, active site is there, okay, and complementary shape. You can see as a pH is increased for above the optimum, see the shape of the active site is changed. And then, this substrate will not fixed here. Now in my enzyme investigation, this is the last slide. Okay. Uh, Normally, like, sometime this type of question, is given in the paper that you can see here. Amylase is an enzyme that digests starch. Okay, are polysaccharides of glucose into maltose, disaccharides glucose. So see, if you just want to test the presence of starch, okay, so either the starch is there or not. Okay, so there is a simple test that is called the iodine test, which we discussed in the previous topic, that is topic biological molecule. That when you put iodine, 2 or 3 drops of iodine, okay, into the sample of food, and if the color of the iodine turns to blue black, it means the starch is there. So starch can be tested for easily using iodine solution. See, starch in iodine, if starch is there, then the blue or black color appears. Okay, it means that starch is there. But suppose that starch is broken down, starch is catalyzed by amylase enzyme, so it means if there is no starch, there is maltose. If the maltose is there, okay, so iodine will the color of iodine will not change. It's going to be remain same, that is, the reddish brown. So iodine doesn't change color. Okay, so this type of investigation, is given sometime, in paper sometime paper 4 or paper 6. Okay, so I hope you understand each and everything. Okay, but still, if you have any problem, any questions, okay, regarding any of the topic, okay, so my WhatsApp number is there, my email address is there, and I'm going to upload this video on the YouTube channel. Also, I'm going to put it on the TikTok, so you can also give some comments. Okay, if you need anything, if you want any other topic to be discussed. Okay. Okay. Thank you very much. Take care. Bye.